Students, welcome to Vivid Botany. We are looking another question from PCR technique. Earlier we have seen a PCR technique question in match the following, but this time the question is very much different. He is asking your ability of understanding the question. We see that in PCR technique, three steps are there. What is PCR? That is polymerase chain reaction. I told you earlier also in my earlier video, polymerase chain reaction. This is discovered by two scientists. One is one scientist, that is Carey Mullis discovered by, and the most important thing is that in PCR technique, if one DNA copy is given, that is made into multiple copies. So it's meant for diagnosis, early diagnosis. It's meant for the harmful mutations. It's meant for cancer genes. It's meant to detect the ELISA, what you call that, early diagnosis of HIV, and even RT-PCR is very much usage nowadays in the diagnostic centers for the detection of corona, that is COVID-19. So here the main thing that I am coming to question here is that in polymerase chain reaction, three steps are there. The one step is denaturation. The another step is annealing. And the third step is extension. So with the three steps, the PCR technique is over. We will try to understand what do they mean exactly. Let me explain you. In denaturation, simply speaking, the temperature is hiked, the temperature is raised. Do remember, denaturation temperature is raised to 96 degrees Celsius, and annealing, the temperature is get lowered. It is lowered down. It's about 55 to 65 degrees Celsius. And in extension, again temperature is raised to an extent of 72 degrees Celsius. Why? Why temperature is being raised? Why temperature is lowered? And why temperature is again raised? It is something like this. This is temperature raising, then temperature is lowering, again temperature is raising. So this is, I'll take once again, this is temperature is raising, and again temperature is lowering, and again temperature is raising. So this is 96 degrees Celsius, this is 55 to 65 degrees Celsius, and it is 72 degrees Celsius. Now the question arises, why we are raising and why we are lowering the temperature. See, we should raise the temperature to separate the two DNA strands. We know that DNA is double-stranded. That is, double-stranded DNA will convert into two separate molecules of single-stranded DNAs. What happens? Why do we convert double-stranded DNA to two single-stranded DNAs? See, if you don't convert dsDNA into ssDNAs, then the two DNAs cannot act like templates, and the two DNAs cannot form the new other two strands. So to make them to act as templates, and number one, number two, and to allow them to act as templates, the two DNA strands need to be separated. At what temperature they get separated? At 96 degrees Celsius they get separated. Remember, at 96 degrees Celsius also DNA remains alive. Only hydrogen bonds are broken down. The glycosidic bonds, ester bonds, diester bonds are not broken down, and DNA is not cleaved, is not fragmented into deoxyribonucleoside phosphates. It is just the hydrogen bonds are breaking down, and the double-stranded DNA converts into single-stranded DNA to act like template during the denaturation process, right? This is the one thing. And second step I told you, annealing. What happens in annealing? In annealing, we attach the primers. Attachment of primers. When we reduce the temperature, primers are attached. What are primers? Primers are small fragment of oligonucleotides that are attached at the, what do you call that, third end of the template strand. For example, if these two are the template strands, let us suppose these two are template strands. Let us, this is 5 to 3 and this is 3 to 5. Now the two strands act like template. So in this strand, here the primer is attached and here the primer is attached. So at what ends we are attaching the primers? We are attaching at third end of the template. This is template. This part of DNA is called template. This is also called as template. So the two DNA strands act as template to which third end of DNA the primers are attached. When do primers are attached? When the temperature is lowered down to 55 to 65, then primers are attached. So attachment of primers at the template strand at third end is called as annealing. Then what is extension? Extension is again raising temperature. So why should we raise temperature? So here TAQ polymerase, I told you it is obtained from Thermus aquaticus. It is a DNA polymerase. This TAQ polymerase will extend the primer. It will extend the primer. See, I am drawing here. It will extend the primer in 5 to 3 direction. So TAQ polymerase extends primer in 5 to 3 direction. See, the 5 to 3 direction of new strand, not of the template strand. So hope it is very clear here. It is very clear like this here. In 3 steps the DNA gets multiplied and 3 steps get repeated. The 3 steps get repeated, like in 30 cycles 1 billion copies are made. So the question is here, when you raise temperature, which process is affected? See, the denaturation process is affected here, because at high temperature the denaturation is only affected. The first step is only affected, and we know that denaturation is meant for separating the two strands of DNA. Okay, I hope my explanation is helpful for you, and try to follow the channel, try to understand all the questions which are very typical, and I am analyzing the questions of two kinds on the same topic. So not to forget to subscribe my channel Vivid Botany. Thank you.